Questions 12 to 17 in the ACER purple paper. Questions 12. This cell is A. Prokaryotic, B. Eukaryotic, C. Undergoing mitosis, and D. Undergoing meiosis. So the most important thing to know to answer this question is the difference between a prokaryote and a eukaryotic cell. In general, eukaryotic cells are more complex cells. They're the cells that are found in the human body, whilst the prokaryotic cells are much more simple, and they're also mostly found in simpler organisms such as bacteria. So I've got a table of a couple of the differences between prokaryotes and eukaryotes. It would be probably good just to know a couple of them before you go into the GAMSAT, just in case you get a question like this. So I'd probably look up and make sure you comprehensively know all the differences, not just the ones on the table, but any other ones that you can find in your own time. But the main ones are here. So we've got prokaryotes, they have circular DNA versus the linear DNA that eukaryotes have, floats around freely. The main difference is that prokaryotes don't have a nucleus versus the eukaryotes, which do have a nucleus to contain all that DNA. Prokaryotes also divide by binary fission, whilst eukaryotes divide by meiosis, mitosis. Prokaryotes are usually unicellular, eukaryotes are usually multicellular. And the important one for answering this question is this one. So prokaryotes are non-membrane bound organelles, whilst eukaryotes have membrane bound organelles. Now, if we look at the figure in questions 12 to 17, the figure of that cell, we can see that all of those organelles have membranes going around it. So our ER, endoplasmic reticulum, that bit where all of those amino acids are feeding into on the left, well, that is membrane bound. So in a prokaryote, what would happen is ribosomes, which are useful for protein synthesis, they sort of just float around in the cytoplasm of a prokaryote. But in eukaryotes, they are mostly located on those endoplasmic reticulum, specifically the rough endoplasmic reticulum, and they're sort of shown in the diagram. They're those parallel stacks of membrane which have those little dots on the surface. Those dots are the ribosomes. So we can see that this cell has membrane-bound organelles and therefore is a eukaryotic cell. So for question 12, B is the correct answer. Question 13. Which of the following is most likely? This cell is in A. Interphase, B. Prophase, C. Metaphase, D. Telophase 2. So answers B, C, and D, they are all examples of meiosis phases. So we need to figure out what is most likely. Well, the reality is that in our cell cycle, if we just draw out a circle here to represent the cell cycle, only a very small portion of that cell cycle is concerned with meiosis, mitosis. So the vast majority of the time, the cell actually exists in interphase. So this part is where all of those stages in mitosis occur. So prophase, metaphase, telophase. That's all within just that very small period of time, whilst most of the time the cell is in that interphase form. Because the cell is not extremely functional when it's going through mitosis/meiosis. Say if you rely on those cells for a specific function, well, when it's constantly splitting up and preparing for replication, it's not going to be the most useful cell. So we actually want our cells to be in interphase most of the time, because that's when the cell actually gets its work done. Where that's when it actually functions and performs those functions. So, which of the following is most likely? The cell is most likely going to be in interphase, and mostly because interphase takes up the most amount of time in the cell cycle, and also because there's nothing else in that figure to indicate that the cell is undergoing mitosis / meiosis. There's nothing to indicate that it's in prophase, metaphase, or telophase. So for question 13, A is probably the correct answer. Question 14. Consider the following experiment. Chromaffin cells were exposed to an antibody that binds specifically to a protein that is located uniquely on the inner surface of the chromaffin vesicle's membrane. The antibody was labeled with fluorescent dye. After exocytosis and then endocytosis, fluorescence would be greatest on the. So we've got our chromaffin vesicle here, and that's this whole pathway is taken directly from the figure. So our chromaffin vesicle is going to be labeled on the inner surface with a fluorescent dye. So I've just got it in blue here. So we're just going to label that inner surface. Now what happens is when it undergoes exocytosis, it makes contact with the cell membrane, and the vesicle merges with the cell membrane. So what's going to happen is the inside surface of the vesicle now becomes the outer surface of the plasma membrane. So the fluorescence now will appear on this side. Now just transferring across this diagram to this one. So again, our dye is still on that outer surface of the plasma membrane, even when it's undergoing endocytosis. So going through that process of endocytosis, i.e. The process of taking in small particles from outside the cell. So exocytosis is used by the cell to expel particles from inside the cell to the outside, the world, I guess, the outside area. So continuing on, the fluorescent dye is going to be located on the inside surface of these vesicles, just like before. So nothing has really changed there. So let's just fill this out for each of the vesicles. Now, the vesicle has sort of one of two options. It can either get engulf/merge with the lysosome, or it can merge with the Golgi complex. So in the diagram, we've got this engulfing situation going on with the lysosome. So the vesicle has been completely engulfed by the lysosome. What sort of isn't really clearly shown is that the vesicle can actually merge with the lysosome. So we're going to get a similar situation as with our Golgi complex that we'll see later, but basically the vesicle will merge with the lysosome, so the fluorescent dye will appear on the inside surface of the lysosome. But we'll stick to what's sort of being shown in the diagram at least so far. And anyway, when we combine the vesicle with the Golgi complex, what will happen is this fluorescent dye will appear on the inside surface of the Golgi complex. So let's go through each of the answers in 14. After exocytosis and then endocytosis, fluorescence will be greatest on the inside of the cell membrane. Well, that's not true. The fluorescence would be greatest on the outside of the cell membrane, the outer surface, not the inner surface. So let's move on. B, inside the Golgi complex. Well, that looks pretty good because we've got that fluorescent dye on the inside of the surface as shown by our little diagram here. So when it merges, the inner surface of the vesicle becomes the inner surface of the Golgi complex when they merge. C, outside of the lysosomal membrane, that's not going to be true. So even if it did merge, we'd expect to see the fluorescent dye on the inner surface of the lysosome, not on the outer surface. And D, outside the mitochondrial membrane. Well, the mitochondria is not even represented in this pathway, so there's not really any way for the dye to get to the mitochondria anyway. So therefore, for 14, the correct answer is B. Question 15. Synthesis of pre-pro-enkephalin is achieved by A. Translation at the ribosomes. So if we can just locate pre-pro-enkephalin on our diagram, it's in that top left-hand corner, just that sort of line just above the endoplasmic reticulum. So pre-pro-enkephalin, it appears to have what's called a signal peptide attached to it. So a signal peptide is a short peptide sequence that is attached to certain types of proteins that are destined for the secretory pathway. Now, the secretory pathway is basically a pathway which leads to the exocytosis of a certain molecule, in this case protein. So this protein, therefore, since it has a signal peptide, is destined for the secretory pathway and is destined to be moved outside of the cell. What's important though is that signal peptides are usually attached to proteins, to peptides. So we can say that pre-pro-enkephalin is a peptide. So therefore, A is correct, because all proteins are, in the human cell at least, are translated at the ribosome. So translation is the process of going from the mRNA to a protein, and usually this all occurs at the ribosome. So therefore, question 15, A is the correct answer. Answers C and D are wrong because the signal peptide is not the precursor for transcription or translation. So in C, we've stated that synthesis of pre-pro-enkephalin is achieved by transcription of the signal peptide. Well, the signal peptide is a result of translation. It's a product of translation, but so therefore transcription of the signal peptide does not occur. And D, so translation of the signal peptide. While the signal peptide is not being translated, it is actually the product of translation. So therefore, D is incorrect as well. Question 16. According to the figure, the mitochondrion. Alright, so unfortunately the figure does not very clearly show whether the mitochondrion is obtaining or storing energy. But if you know a little bit about the mitochondrion, even just the very basics, you should hopefully be able to answer this question. So the one sort of fact that most people know about the mitochondria is that the mitochondria is the powerhouse of the cell. So if we are the powerhouse of the cell, we're going to be producing energy, right? So therefore, we wouldn't expect the mitochondria to be obtaining energy from other things. It's not sort of burning up / using energy from another source. It's actually the source of power itself. It's going to be storing energy in molecules, particles, whatever. So we can already say that for question 16, A and B are wrong because the mitochondria is the powerhouse of the cell. It is going to be the place where we're making energy, not using it. So we're left with C and D. C, we state that the mitochondria stores energy in bonds in ATP. And in D, we say that the mitochondria stores energy in bonds in ADP. And to figure this out, well, we just have to look at our figure. We go from adenosine to AMP, from AMP to ADP, and finally from ADP through the mitochondria into ATP. So what we're saying is that we're going from ADP to ATP before and after the mitochondria. And since we know that the mitochondria is storing energy in whatever particle goes through it, when we go from ADP to ATP, we're going to be gaining energy. Therefore, we're going to be storing energy in bonds in ATP. So, therefore, the correct answer for question 16 is C. Question 17. Which one of the following is a precursor of adrenaline? So, firstly, it's important to just locate adrenaline. So, adrenaline is located in one of those chromaffin vesicles. It's at the sort of in the middle right-hand side of the diagram and is represented by a little circle. So, adrenaline, let's just work back and figure out what is sort of converted into it. So, adrenaline is that circle. If we follow that arrow back down to the curved arrow, well, what we're saying there is we're going from the square is noradrenaline, to the circle, adrenaline. So, noradrenaline is a precursor, therefore, for adrenaline. Going back up the pathway, so noradrenaline is converted from dopamine, dopamine is converted from dopa, and dopa is converted from tyrosine. So, therefore, for question 17, which one of the following is a precursor of adrenaline? Or A, we've got tyrosine, as we just stated, tyrosine is a precursor for adrenaline because we worked through that pathway. So, therefore, A is the correct answer for question 17.